Hello everyone. I hope it has been a great day so far for you, no matter where you are. My name is Wei Pin and I'm from the Universities of Leeds. First of all, I would like to thank Amalia for inviting me to present my research work here, and my thanks also extends to Avery for hosting this webinar. My talk today is about a piece of work that I did a few years ago on the development of a scaled-down agitated filter bed dryer for assessing particle breakage in the pharma sector. The work that I'm presenting today is actually a part of my PhD studies looking at the attritions and transformations of pharmaceutical crystals during agitated filter bed drying. My talk will be focusing on the first two items here. I'll first go through the development of a lab-scale attrition tester for agitated filter bed drying that is built on top of a FT4 rheometer. Following that, I will then show you a case study looking at evaluating the breakage of a pharmaceutical ingredient, carbamazepine dihydrate, using the rig that we developed. For those of you who are not familiar with the agitated filter bed drying process, it is actually the unit operation of choice in the pharmaceutical industry for isolations of potent active pharmaceutical ingredients. Following filtration, heat is normally supplied to the wet cake and an impeller is agitated intermittently to enhance the heat transfer and promote homogeneous drying of the drug substance. Dry nitrogen gas is often applied under positive pressure to push out any unbound solvent from the wet cake. The shear deformation happening during this process could actually result in particle breakage, agglomerations, polymorphic changes, and also some process-induced disorder of the API. This is often very undesirable as it could lead to further downstream processing issues such as degraded product qualities and deviations from the product specification. Agitated filter bed dryers vary in scale and could accommodate product mass from a few kilograms to hundreds of kilograms. A typical AFBD consists of a jacketed vessel with a preferred material of constructions, normally HIS alloy, and second of all, a drive assembly with a heated agitator system capable of rotational and axial movement. We would also have a heated filter plate with interchangeable filter media, a dust filter, and also some side discharge and sampling valve. Here I've borrowed an online video from Heinkel Group just to show you what is happening during an agitated filter bed drying process. It normally consists of filling the system with a slurry of crystals and solvent. The filtration process will then drive out the unbound solvent, after which the wet cake will be washed and rinsed a few times before drying it. During the drying process, the impeller may be rotated intermittently and also programmed to induce axial mixing by going upward and downward as per need basis. Once the crystals are fully dried, they then get discharged and a cleaning protocol will start. As I mentioned earlier, the filtration process removes the muddy liquor from the crystallized products. A number of washes are then applied to flush out the impurities using a wash solvent that has similar composition to the muddy liquor. In some cases, a wash solvent having different compositions can also be used when it proves beneficial to do so. For instance, to reduce agglomerations and for better purging of impurities. Once the muddy liquor is filtered and the wet cake is settled, a vacuum or headspace pressure can then be applied to deliquor the excess unbound wash solvent. During this process, smoothing of the wet cake using the impeller may be performed, and typically we're talking about 3 to 5 impeller rotations to fill in any cracks formed on the free surface, which could help avoid the bypass of nitrogen flow and increase the efficiency of solvent removal. However, the vacuum itself will not remove all the solvents in the wet cake. Quite often, we need to heat up the wet cake to the upper temperature limit, and that is decided by the thermal stability of the API. The heating rate is governed by the loss of drying of the solvent contents and the propensity of hardening of the wet cake, which is very likely to happen when we have a very fast drying rate. So by applying nitrogen gas, we can actually help to speed up the vapor removal, either through headspace purging or through the wet cake. And in some cases, the drying can be done under controlled relative humidities and temperatures to help achieve the desired crystal forms. When we get to the desired drying temperatures and loss of drying, the impeller is then agitated to help accelerate the drying. The typical speed that we use in industrial operation is approximately 5 to 10 RPM. For materials that are prone to breakage, we can go as low as 2 RPM. But of course, the speed used is dependent on the dryer size as well. For a typical dryer with a diameter of 2 meter, the maximum tip speed is around 2 meter per second. During the agitation, heat can also be supplied through the impeller as well as the bottom plate. The intermittent mixing can be continued until an acceptable levels of solvent content is achieved according to the ICH standards or product specification targets. This can take from hours to days, depending on the API physical properties to be controlled, the crystal form, and also the type of solvent used. Well, I hope now you have a better understanding of the agitated filter bed drying process. As you can see, it is not an overly complicated system, but it does involve a good number of steps. And given the scale of the system and the time needed to dry a product, it is important to have a prior knowledge of how the product will behave in the dryer. One way to do it is by testing the system in a smaller scale, but unfortunately commercial lab scale AFBD is not widely available. There are a couple of test devices reported in the literature, but then again they are custom built and available in-house only. Our idea for this work was to build an AFBD unit that could actually sit on a common test device, and the sole test device is the Freeman Technology FT4 rheometer. This work was a development program established between the University of Leeds and Freeman Technology. The FT4 rheometer is an ideal surrogate test device as it has a very similar setup compared to an AFBD. What makes it even more attractive is that it can measure the torque of the impeller and have a flexible control of the motions of the blade. When we design the new test rig, these are the requirements that we have in mind. It needs to have an interchangeable impeller blade geometry to assess the efficiency of blade design in promoting homogeneous drying. We also need a system where we could have a variable impeller rotational speed. In addition to that, a programmable impeller motion for devising optimal protocols for drying is also needed. Drying is important, and therefore the new system needs to have a heating capability for assessment of drying at elevated temperature. Filtration capability with high chemical resistance is essential, together with vacuum capability for isolations of products from waste liquor. And last but not least, the new system will come with an array of sensors probing the temperature, pressure, and humidity at various stages during agitated filter bed drying. As I mentioned earlier, the FT4 rheometer is an ideal surrogate device for our AFBD purpose. One, that's because it's a commercial flow characterization device widely adopted in various industries. And two, it has high precision torque measurement and impeller motion control. We make use of the standard vessels of the FT4 rheometer, measuring at 25 mm in diameter. The impeller blade diameter is about 23.5 mm, hence making the impeller wall clearance to be about 0.75 mm. The vessel is jacketed so that heat can be applied to the system to accelerate the drying. With this system, the rotational speed can go up to 120 RPM, which is ideal for our application. The blade geometry is a functional part of the AFBD as it is used for smoothing and ploughing of the wet cake during the drying process. The standard FT4 blade unfortunately is not designed for this purpose, but we have made it possible to replace the impeller blade with a design of interest. What we are seeing now are actually the scaled down versions of a AFBD blade. Right at the middle, we have the blade which was made by 3D printing, and the one on the right is made via CNC machining. The difference is that the 3D printed one have a rougher surface, while the CNC machine one is smoother. The AFBD blade has a very intricate design. It has an angle base with an angle of 15 degrees, and the blade inclination is about 60 degrees. It is actually an exact replica of a conventional AFBD blade, but somehow scaled down to fit into the FT4 rheometer for our purpose. The FT4 rheometer offers the flexibility to program complex impeller motions. Using it, we are able to reproduce standard procedure in AFBD, such as smoothing, plowing, and intermittent agitation in the prototype. Just to show you some videos of the new test rig in action, on the left we have the impeller rotating clockwise, which gives a smoothing effect, and on the right the impeller is rotating anti-clockwise, which simulates the plowing and agitation process of the agitated filter bed dryer. The impeller blade design is directional in a sense that the directions of the impeller rotation will give rise to different functions. In our new test rig, we incorporated a wide array of sensors to monitor the conditions of the AFBD process, such as the ambient temperature, pressures, and relative humidity. We also have a set of sensors looking at the temperatures of the vessels, the jacket, and also the outlet. Of course, we are also monitoring the impeller torque and the normal load applied to the system. The current slide is showing an overview of the system that we have put together. The photo on the left bottom is showing the new AFBD test device that we designed, and it can be sit on top of the FT4 rheometer, as shown in the photo to the right. Essentially, the new system will be connected to a vacuum pump for filtration, a water pump to circulate a hot fluid through the jacket vessel, and a water bath which regulates the temperatures of the circulating fluid. We also have a Raspberry Pi which acts as a data acquisition module, and it is connected to a computer for exchanging the data gathered from the sensors. Well, this is a video showing the AFBD in actions. So we have the FT4 software, as you can see here, which is connected to a Raspberry Pi for collecting the sensor data. And the FT4 rheometer is connected to a vacuum pump for filtrations. As you can see here, we have the test rig that we've designed, which is in action now, and it is connected to a mini water pump and a water bath which circulate the hot fluid. Let's take a look at a quick study to simulate an agitated filter bed drying process using sodium chloride crystal and isopropyl alcohol as the liquid carrier. This is a plot showing the average torque measure as a function of the number of times the impeller is inserted into the powder bed and rotated. This result was collected with vacuum applied, and we can see that at first a high torque is measured, but it quickly levels off due to the quick drying of the IPA solution in the presence of vacuum. Now, if we were to repeat the same process without the vacuum, we get a very nice curve which shows three different regimes of the drying process. At first, when we have a high volume of IPA in the system, the mixture behaves like a slurry in which the solvent acts as a lubricant, hence reducing the resistance to flow. As the IPA dries off, the mixture then gets to a state where the capillary force becomes prominent, and that increases the resistance to flow. As the drying progresses, eventually all the IPA evaporates and only the sodium chloride crystals are left, which are free-flowing. So essentially what I'm trying to demonstrate here is that our system is able to pick up the tiny change in the particle dynamics during the drying process just by monitoring the change in the impeller torque. And this is just a plot showing the change in conditions monitored by the sensors we deploy in our test rig. You can see how they change as a function of time. Our interest is in particle breakage, and here I'm just showing you what happens to these sodium chloride crystals after the drying process. We can clearly see an evidence of particle breakage here, as we can see that changing the rotational speed of the impeller does not actually have much effect on the extent of particle breakage, at least visually. On the other hand, increasing the number of impeller motions has an obvious effect on the extent of particle breakage. We can see that at 4,800 revolutions, the particles broke into smaller pieces compared to that of the 300 revolutions. So, what can and cannot be done in the current prototype? We can change the impeller design, we can program the impeller motions, we can control the temperature of the jacketed vessel, we can also do filtrations and apply vacuum, and we can monitor the ambient conditions. The only exception is that we cannot apply any head space pressure, as that would involve a more sophisticated design so that it would not interfere with the torque measurement. Just some interim remarks here. I hope I have demonstrated to you that our new test rig is capable of running all the basic functions of an agitated filter bed dryer through the quick study with sodium chloride and IPA. You can see that the FT4 rheometer is able to capture the change in the powder bed as drying progresses in the new test rig. Now that we have a working prototype, let's move on to a more systematic study on the evaluations of attritions of carbamazepine dihydrate crystals due to agitated filter bed drying. In this work, we will be subjecting the crystals of interest to different process conditions and measure the shift in particle size distributions using an optical imaging technique. The shift in the particle size distributions will be used as an indicator to infer the extent of breakage and later on correlating with the input energy. The process parameters that we studied here include the impeller clearance from the base, impeller tip speed, impeller angular displacement, solvent content, and the use of vacuum. The procedure is quite straightforward. We first subject the feed crystals to different process treatments. We then collect the products and measure the size distributions and shape distributions. Carbamazepine dihydrate is the hydrate forms of carbamazepine, which has two water molecules embedded in its crystal lattice. The presence of these water molecule channels in fact constitutes to the cleavage planes of the crystals, which are parallel to the OKO phase of the crystals. These planes are crystallographic planes of weakness, meaning the crystal tends to cleave or break along these planes. What we are seeing here are some of the SCM images of the carbamazepine dihydrate crystals. It is very clear that they are of high aspect ratios and they are very platy. The dominant phase is the HOO phase, and we can see the cleavage planes that are perpendicular to the dominant crystal phase. Here we can see the initiation of crack propagations along these cleavage planes as induced by the vacuum inside the SCM. As the water evaporates from the crystals, the cracks form on the surface of this crystal. Under normal conditions, this crack would not be visible, and the interesting crystal structures and the presence of cleavage planes of this compound makes it a very nice system to study in our new test rig. As the morphological informations of the crystals are of interest here, we opted a particle size analysis system based on optical imaging technique. The morphology G3 of Malvern Panalytical is ideal for our application as it measures two-dimensional informations of the particle size and shape. Particles are first dispersed onto a glass slide, the particles on which will then be captured by the microscope and have their size and shape information analyzed. The result of this device is normally presented in the form of volume distribution, in which the volume of particles are calculated based on their projected area on the glass slides, assuming a volume equivalent to a sphere. However, that assumption is inadequate for our system as it overestimates the volume of plated particles like carbamazepine dihydrate. In order to correct that, we define the volume of plated crystals to be proportional to their projected area under the microscope, multiplied by their thickness. The variation of this thickness is assumed to be very small and hence also assumed to be a constant. Having that, we then move on to the definitions of extent of breakage. The extent of breakage R star is conventionally known as a ratio of the mass of debris to the mass of the fit crystals. If we now assume the particles are non-porous and have the same density, this gravimetric ratio can then be converted into an area-based ratio as shown here, where R star is simply a ratio of the area of the debris over the area of the fit crystals. And the way we get this fractional loss is through subtracting the particle size distributions of the broken crystal from the particle size distributions of the fit crystals. See the red shaded region of this plot. The area of this plot will actually give us a measure of the shift of particle size distribution and an indicator of how much the particles have broken. And these are the mathematical formulae that we use to calculate the shaded region, but I won't be going through them in detail here. To calculate the extent of particle breakage, we first need to measure the fit particle size distribution. As shown in this plot are the distributions of the length and width of the carbamazepine dihydrate crystals. Here we also introduce a new size parameter known as the square area equivalent side length, which is a measure of the particle size based on their projected area, equivalent to that of a square. The characteristic sizes D10, D50, and D90 are also shown in the table here for your reference. Let's have a look at the first process parameters that we study in this work, the clearance size. As you all know, shear deformations of crystals is most prominent at the region between the impeller blade and the vessel base, where the shear bend actually prevails and the clearance is highly influential on the stresses experienced by the particles within this tiny gap. Too small the gap, the particles could actually undergo extensive particle breakage due to crushing. So 3 different clearances of 1, 3, and 5 millimeters were tested in this work. The time and speed of the impeller rotations are kept at 40 minutes and 120 RPM respectively. The ratio of particle length to clearance are about 2.8, 8.5, and 14.3 particle length for the clearance sizes of 1, 3, and 5 millimeter respectively. So what we find interesting here is that the change of the clearance size has only minor influence on the extent of particle breakage. The extent of particle breakage obviously increases as the gap is made smaller. As you can see here, it is increased from 5% for 5 millimeter gap to about 9% for 1 millimeter gap. The smaller the clearance size, the larger the extent of breakage. The difference in R star, however, between the 3 and 5 mm clearance sizes is very small compared to that of 1 millimeter, indicating that the particle breakage of both of these cases are actually due to the shear deformation of the powder bed induced by the impeller and not by crushing. So what we learn from this result is that selecting an appropriate clearance size is critical in minimizing unwanted breakage of particles, but we need it to be small enough to promote a homogeneous drying across the powder bed during agitated drying process. The second process parameters that we studied here is the impeller speed. While keeping the number of rotation constant, 3 different impeller rotational speeds of 30, 60, and 120 RPM were used to agitate the particle bed, and those are corresponding to the impeller tip speed of about 0.04, 0.08, and 0.15 meter per second. Of course, these are lower than the typical tip speed used in an industrial scale agitated filter bed dryer, but nevertheless, they provide a comparative studies of the sensitivities of the model crystal breakage to strain rate. So it was found in this work that for the range of speeds used here, the extent of breakage of carbamazepine dihydrate crystals were not sensitive to the impeller speed. Increasing the impeller rotational speed by 4 times only increases the extent of breakage by 3%. Agitations at 60 rpm, it causes the highest breakage extent at 9.5%, but when compared to the other two impeller speeds, the difference is really not significant. Depending on the nature of the materials, some powders are highly sensitive to strain rate and could result in a lot of breakage. The ability of the current AFBD setup to study the powder behavior under different strain rates is highly desirable, providing useful information for developing drying protocols that are best suited for different powders of interest. Next, we also study the effect of agitation time. Carbamazepine dihydrate crystal were subjected to 4 different lengths of agitation time, 5, 10, 15, and 20 minutes, while keeping the rotational speed constant. Here we can see a very nice dependency of the extent of particle breakage on the agitation time. Increasing the agitation time basically increases the extent of particle breakage, as is nicely shown in this plot. In industrial operations during the drying stage, the system is closed and the application of vacuum to the vessel generally does not cause increased compressions on the powder bed. However, the wet cake could be compressed and consolidated due to its own weight, as well as by pressure differentials across the bed during the washing cycles. Because of the smaller scale of our test rig here, the stresses experienced in the laboratory scale setup are significantly lower than the corresponding industrial case. In order to artificially increase the compressive stresses to simulate industrial conditions, vacuum suction is applied here. In this work, a vacuum pump was connected to the bottoms of the AFBD vessel to investigate the effect of vacuum suction's applications on the breakage of carbamazepine dihydrate crystals. Depending on the particle size and bed packing, the pressure drop across the powder bed can vary. And applying vacuum suction to the bottoms of the AFBD vessel imposes additional normal load to the powder bed due to the pressure drop as the air percolates through the packed bed. The first thing that can be noticed in the results here is that the extent of particle breakage is much higher compared to the same set of experiments performed without vacuum suction. It's very clear that at the clearance size of 1 millimeter, the resulting extent of breakage is as high as 41%. The extent of breakage is reduced as the clearance size is increased, going from 2 to 3 millimeter. A large reduction in the extent of breakage of about 20% is observed. The R star at 3 and 5 mm clearance is about 17 and 14% respectively. So what is telling us is that there may exist a critical clearance size somewhere between 2 and 3 millimeter, below which particle interlocking causes extensive breakage. So above this critical clearance size, the effect of clearance size diminishes, and the R star does not vary that much with further increase in the clearance size. All the results that I've shown so far were all done on dry powder without the presence of solvent. As I've shown previously in the sodium chloride work, the presence of liquid in the powder bed could drastically change the resistance of the powder bed to flow. In other words, the interparticle friction also changes due to the presence of liquid. In this work, an aqueous slurry of carbamazepine dihydrate and water was first prepared. The slurry was then subjected to filtration in the AFBD vessel until a desirable solvent content was reached. 3 solvent contents were tested here in this work, the 10, 25, and 50%. The wet cakes with different liquid contents were then agitated at 120 rpm with an impeller-based clearance of 2 mm to assess their role in the breakage of carbamazepine dihydrate crystals. No vacuum suction was applied here. The total agitation time was 5 minutes, and after that the sample was left in the vessel with vacuum suction applied so that they are air dried at room temperature for 2 hours before we measure the particle size in morphology G3. It is evident that the solvent content is influential on the reduction of particle size here. The higher the liquid content in the powder bed when it is being agitated, the higher the extent of breakage of the crystal. At 10% liquid content, the extent of breakage is about 12%, and it increases to 32 and 37% when the amount of water in the powder bed is increased, corresponding to the liquid contents of 25% and 50% respectively. Now, this is expected as a decrease in liquid contents in the powder bed reduces the attraction force between the particles, which in turn leads to less resistance of the powder bed to flow when agitated by the impeller. These results have shown that the optimal time for applying agitation is when the cake is sufficiently dry. Applying agitation can actually cause severe breakage of the particles, and it should be avoided. It is also worth noting that beyond a critical point, at high liquid content, the liquid content could in turn, like now act as a lubricant instead and reduce the bulk frictions of the powder bed, as I mentioned earlier in the previous sections. In this work, we were also interested in understanding how the carbamazepine dihydrate crystal break during agitated filter bed drying. The change in the crystal shape due to breakage under shear deformation was characterized by image analysis. The reduction in width and length expressed in their characteristic particle sizes at D10, D50, and D90 is shown in this slide. So there are three series of data points here. The blue one corresponds to D10, orange corresponds to D50, and gray corresponds to D90 of the size distributions of the length and width of the broken particles due to different process treatments. Each data point here corresponds to a set of process conditions tested in earlier sections, and the correlation between the length and width of the broken crystal subjected to different process treatments shows a strong linear relationship between the characteristic sizes of the length and width distributions. The slope of the regression line of each data series gives us a measure of the average aspect ratios of the broken crystal. D90 is associated with the largest particle group in a distributions, while D10 represents the smallest particle group. The slopes of the regression lines are found to be 0.39, 0.49, and 0.54 for D10, D50, and D90 respectively. So there's a trend of reducing aspect ratio, as we can see here, with the particle size, but it is not much, even though the crystal have been subjected to substantial breakage. So what this is telling us is that there are two breakage mechanisms at play here. First of all, fragmentations could happen during this process, in which the crystal break perpendicular to their largest dimension, snapping them into smaller pieces. The breakage could also due to cleaving of the crystal along the crystallography planes of weakness, which are parallels to their largest dimension. And as a result of these two mechanisms, and the change of aspect ratio is not as significant as we expected. Just to show you some of the images of the broken crystals, in this slide we are looking at the broken crystal, which are bigger in size, and we can see that the big crystals are generally very wide, but not so long in terms of their length. In other words, the aspect ratios of this large crystal is not that high. Conversely, if we now look at the broken crystal, which has got smaller size, it is very clear that those smaller crystals are slender in general, which explains the trend that was observed earlier. Evidently, carbamazepine dihydrate crystal could break either by chipping off the corner or snapping along the longest dimension. With the FT4 rheometer, we can measure the torque of the impeller during the agitated filter bed drying process, and that gives us the ability to calculate the amount of work done by the impeller or the energy that we put into agitating the powder bed. To shear a powder bed, a substantial amount of mechanical energy is needed, and that explains the initial spike of the torque recorded here. As the powder bed starts to flow, the shear deformation induced on the powder bed causes the particles to break. As the particles get smaller in size, their resistance to flow becomes less, and hence why a gradual decrease in the magnitude of the torque is observed here. The input mechanical energy for the experiments performed was calculated, and the relationship between the R star and the specific input energy is drawn in this plot. We can see that a reasonably good fit is obtained from linear regressions, indicating that the extent of particle breakage is in fact proportional to the expended input energy. In conclusion, a new test rig to simulate agitated filter bed drying has been developed in collaborations with Freeman Technology using existing FT4 powder rheometer. The highly versatile device performs the essential features of an industrial-scale agitated filter bed dryer, capable of filtrations, programmable axials and radial mixing, temperature and humidity control, vacuum and nitrogen purge impeller blade customization, and process monitoring via built-in sensors for torque measurement, pressures, and humidity. The new AFBD unit is used to assess the breakage of carbamazepine dihydrate crystals, and being long and platy, the optical image processing and the analysis is used to characterize the shift in particle size and shape. The extent of breakage is calculated by assessing the shift of the size distributions of the broken crystal from the fit crystals. The extent of breakage is increased when the impeller base clearance is reduced. The impeller tip speed does not influence the breakage of carbamazepine dihydrate greatly for a given number of impeller rotations. Increasing the number of impeller revolutions increases the extent of breakage, as intuitively expected. Applying suction to exert cake consolidation increases the extent of breakage significantly, as the resistance to shearing is increased in this process. Liquid content is another major factor in agitated filter bed drying that could cause significant particle breakage. Water was used in this work, and at high liquid content, 50% water, the particles are held together strongly by the capillary bond between the particles, and agitating the powder bed at this stage could actually cause significant particle breakage. And when the liquid content is lowered, the breakage of the crystal is reduced proportionally as the attraction force between the particles is reduced. Carbamazepine dihydrate crystals undergo fragmentation first, followed by chipping when the particles becomes more equant. The mechanical energy expended by the impeller correlates well with the extent of particle breakage. The proposed laboratory scale instrument that I've presented here provides a tool for comparative assessment of the propensity of particle attritions under agitated filter bed drying conditions. The research work reported here was part of a tripartite research program executed by the Universities of Cambridge and Leeds and Imperial College London. This project was financially supported by AFBD Chicago in the United States, and I would like to thank Professor Bill Jones and Professors Jerry Hink for their helpful suggestions. And I would also like to acknowledge the helpful discussions that I had with the AFBD colleagues, and also for their constant support. I am most grateful to Freeman Technology for supporting the development, and would like to thank the team led by Mr. Jamie Clayton for designing and constructing the agitated filter bed drying unit here. Thank you very much for listening, and I'm happy to take any questions that you have.